Hello children, welcome to AIMS India's online classes. This is biology session. You are watching Anatomy of Flowering Plants part 4. In the last class, we have studied about the internal structure of dicot root, monocot root, dicot stem and monocot stem, isn't it? So, before entering into today's class that is about the leaf, dicot and monocot leaf, let us recall about monocot stem, dicot stem and monocot root, dicot root. Okay children. So, if you take dicot root and monocot root in the internal structure, when we compare, the cortex comparatively narrow in dicot root, whereas it is very wide in monocot root. Okay, endodermis, Casparian strips are more prominent in dicot root. They are less prominent or they are visible only in the young roots in monocot root. Next, if you take the pericycle in dicot, it shows it produces the lateral roots and cork cambium and the part of vascular cambium, whereas in monocot it produces only lateral roots. Okay. The vascular bundles, if you take in monocot root, there are 8 or more in number, but in dicot root 2 to 5, very rarely 8. And if you take the pith, either it is absent or very small, whereas in monocot root the pith is very large and well developed. And if you come to the anatomy of stem, dicot stem vascular bundles, they are arranged in the form of ring, okay, whereas in monocot stem the vascular bundles are scattered, and the nature of vascular bundles in dicot stem, they are conjoint, collateral or bicollateral and open type due to the presence of cambium, whereas the vascular bundles in monocot stem, they are conjoint, collateral and closed due to the absence of cambium. The bundle sheath is absent in the dicot stem. Bundle sheath is usually present in the monocot stem, and the phloem parenchyma is present in dicot stem. Phloem parenchyma is usually absent in the monocot stem, isn't it? Pith or medulla, if you take, it is made up of parenchymatous cells situated in the center of the stem in dicot, whereas in monocot it is absent. Pith or medulla is absent in monocot stem. Ground tissue, if you take, it is differentiated into cortex and pith in dicot stem, whereas in monocot stem the ground tissue is not differentiated into cortex and pith. Hypodermis cholenchymatous in dicot stem. In monocot stem it is sclerenchymatous. Endodermis, if you take, it is one layered, starchy, which is usually not well differentiated in dicot stem, whereas in monocot the endodermis is totally absent. Pericycle, medullary rays, secondary growth, these are all absent in the dicot stem, they are present in the monocot stem. So, if you come to the anatomy of leaf, first dicotyledonous leaf, it is dorsiventral leaf, it is also called. The vertical section of dorsiventral leaf shows 3 main parts. They are epidermis, mesophyll cells and vascular system. Okay, what are the parts of the cross section of dicot leaf? It shows mainly epidermis, okay, adaxial epidermis and abaxial, means upper epidermis and lower epidermis. Then mesophyll cells, this mesophyll is present in the middle, So, it is differentiated into spongy parenchyma and palisade parenchyma. Mesophyll is the largest layer, okay, in the leaf. Then vascular bundles, if you take the vascular bundles, the vascular bundles show xylem and phloem, that is vascular tissue. So, epidermis, if you take the first part, upper part, the epidermis covers the both upper adaxial and lower abaxial surface of the leaf, and it has a conspicuous cuticle. Abaxial means upper epidermis, lower epidermis. Abaxial means lower epidermis is particularly denoting the lower surface of the leaf. The surface has more stomata when compared to adaxial or upper surface of the leaf, upper surface of the epidermis. The tissue between the upper and lower epidermis is called as mesophyll tissue. This is the major tissue. Okay. So, this mesophyll tissue upper towards the adaxial surface of the leaf possesses large number of chloroplasts and the less air spaces are present, that is palisade mesophyll tissue or palisade parenchyma tissue, whereas the mesophyll tissue which is towards the abaxial or lower surface of the leaf, it shows more air spaces, that is why it is spongy mesophyll tissue or sponge parenchyma. Okay. Next, the mesophyll cells are of two types, palisade parenchyma and spongy parenchyma. The adaxial, the upper in particular denoting the upper surface of a leaf, placed palisade parenchyma, it is made up of elongated cells. It is made up of elongated cells, the palisade parenchyma, and arranged vertically parallelly to each other. Okay. And the sponge parenchyma, it is made up of oval or round shaped cells, loosely arranged cells below the palisade parenchyma. This sponge parenchyma is present. The vascular bundles are seen in the midrib region. The midrib, if you take a leaf, what is midrib region? So, this is a leaf. So, this is the midrib region. Okay, if you take the section, the midrib region. So, here it denotes the midrib region where these vascular bundles are present. The vascular bundles are surrounded by a layer of thick walled bundle sheath cells. So, these are bundle sheath cells are present. Okay. Next, monocot leaf, monocotyledonous leaf, it is isobilateral leaf. Okay. Whereas a dicot dorsiventral leaf, it is isobilateral leaf in monocots. It is similar with the dorsiventral leaf in many respects, except the stomata are equally distributed on upper and lower surfaces in the monocots, whereas in dicots, in the lower surfaces more number of stomata are present compared to the upper surface. Okay, the mesophyll cells are not differentiated into palisade and spongy parenchyma in monocots or isobilateral leaves, whereas in dicots it is differentiated into palisade and spongy parenchyma. So, in grasses, for example, in monocots, in grasses certain adaxial epidermal cells along the veins modified themselves into large empty colorless cells called bulliform cells. Okay, means the upper surface cells they are modified to bulliform cells. So, these causes rolling of the leaves to reduce the transpiration during the water scarcity or water stress time. Okay, you can see here. So, this is the adaxial upper epidermis region and this is the abaxial lower epidermis region. In both regions, as we discussed, the stomata are present on both surfaces. Okay, the mesophyll tissue is not differentiated into palisade and spongy parenchyma. Okay, example grass you can take. Next, the secondary growth. Before going to secondary growth, dicot, monocot leaves, if you see the difference, the type of leaf in dicot it is dorsiventral, the type of leaf is in monocot isobilateral. Then stomata usually more on lower surface or lower epidermis in dicot, whereas equal on lower and upper epidermis in monocot. The mesophyll, if you take, it is made up of two types of cells, palisade and spongy parenchyma in dicot leaf, whereas in monocots only spongy parenchyma is present, which has very small intercellular spaces. Bundle sheath, if you take, it is made up of parenchyma, but just above and below the vascular bundles some parenchymatous cells or collenchymatous cells are present up to epidermis in dicot leaves, whereas in monocot the bundle sheath of parenchyma, but made up of parenchyma, but just above and below the vascular bundles are found sclerenchymatous cells. And if you take the bulliform cells, they are absent in the dicot leaf, whereas they are present particularly in grasses, they are present in monocot leaf. Okay. Now come to secondary growth. What is secondary growth? Means extra growth. The secondary tissues are formed by two types of lateral meristems, that is vascular cambium and cork cambium. Okay. So let us see what is secondary growth. Apart from primary growth, the dicot exhibit an increase in the girth, means the diameter of the stem. It is called secondary growth. So the tissues involved in the secondary growth are two lateral meristems, that is vascular cambium and cork cambium. Vascular cambium and cork cambium. This vascular cambium is formed from the conjunctive parenchyma and pericycle, whereas cork cambium, it is also called as Phylogen, it is formed from pericycle. First, vascular cambium. The cells of this cambium present between the primary xylem and primary phloem, it is intrafascicular cambium. Okay. The cells of cambium present between the primary xylem and primary phloem is the intrafascicular cambium. The cells of medullary rays adjoining these intrafascicular cambium become meristematic and form the interfascicular cambium. So two cambiums are formed, intrafascicular and interfascicular. Intra means inside, inter means in between. Okay. So here you can see this secondary growth, how it is formed. So this is epidermis, cortex, primary phloem, vascular cambium, primary xylem, and this is interfascicular, means in between the cambium of vascular bundles. This interfascicular cambium is formed during the secondary growth. Okay. So next here you can see the phylum and phylogen. Phylogen means cork cambium, it is nothing but. So these medullary rays, secondary xylem. So here primary xylem you can see, but during the secondary growth, secondary xylem, secondary phloem and the cambium rings are formed. Okay. So this is due to the secondary growth, the wood is formed in the dicot plants. Okay, we can count the number of rings as annual rings. The age of the plant also you can estimate by counting these rings. Next, the intrafascicular cambium and interfascicular cambium join to form a complete cambium ring. The cambial ring becomes active and begins to cut off new cells both toward inner side and outer side. So this phylogen is a couple of layers in thick. The thickness of this phylogen is 2 layered in thickness and made of narrow, thin walled and nearly rectangular cells. It is made up of rectangular cells, and the phylogen cut new cells to both inner and outer side. The outer cells differentiated into cork or phylum. So this phylogen outer cells will modified or form the phylum, and the inner cells differentiated into secondary cortex or phyloderm. Secondary cortex or phyloderm. Okay. Next, the cork is impervious to water due to suberin deposition, the chemical substance, the cell wall made up of suberin here. The suberin deposition will prevent the water to enter inside. Phylogen, phylum and phyloderm are collectively known as periderm. Okay. So, what is periderm means the cork cambium produces phylum. Phylum is also called cork cells on the outer side and phyloderm on the inner side. Phylum, phylogen and phyloderm together form the or together constitute the periderm. The bark is a non-technical term. Bark, it is the outermost layer of the stem. Okay, it can be peeled out. It is very thick. Bark is a non-technical term that refers to all tissues exterior to the vascular cambium. So, therefore, including the secondary phloem and periderm. Okay. So, actually the balloon like structures are present here. So, there are certain loosely arranged areas in the periderm formed due to rapid activity of phylogen. They are called lenticels. You heard about the lenticels. Lenticels help in gaseous exchange just like stomata and also involve in transpiration in higher plants. Okay, and tyloses. Tyloses are some balloon like structures. They are produced due to the ingrowth of xylem parenchyma into the lumen of xylem vessels through pits. Okay, the bark is all the tissues outside vascular cambium. Bark is all the tissues which is outside the vascular cambium. At certain region, the phylogen cuts off parenchymatous cells inside the cork. So, these parenchymatous cells ruptured the epidermis and forms lens shaped openings called lenticels. How these lenticels are formed? So, these are parenchymatous cells, they are ruptured the epidermis and forms these lens shaped cells or openings called lenticels. So, these lenticels help in the transpiration and exchange of gases between the outer atmosphere and internal tissues of the stem. Okay. Next, secondary growth in roots. So, in you can say the wood formed in the single year, 1 year, it is called as a annual ring, and this can help in determining the age of the tree. Spring wood, it is also called early wood. It is the secondary xylem formed during the spring when the cambial activity is more. So, these wood elements are larger in size and have wider lumen. Another wood by name autumn wood. This autumn wood is also called late wood. It is the wood element formed during the winter when the cambial activity is less. So, the wood formed is lesser in amount and have very narrow lumen. The wood which is formed during winter, it is very less and have narrow lumen. Okay. In perennial woody trees, the region which consists of dead elements and do not conduct water but provides mechanical support is called as a heartwood. Another type of wood that is called heartwood, or it is also called duramen, and the outer or peripheral portion is very soft and lighter in color, constituting of living cells, and it is called sapwood. Sapwood means it is also called alburnum. Okay, and it helps in conduction of water and minerals. The wood of gymnosperms, if you take it is called as non-porous or soft wood, okay, due to the absence of vessels and fewer fibers, and that of the dicots it is called as porous wood, or it is also called hardwood. In dicots it is called hardwood, which contains the vessels and abundant fibers. Okay. This is about the secondary growth in stem. If you come to the secondary growth in roots, in dicot root the vascular cambium is completely secondary in origin. So, it originates from the tissues below the phloem, a portion of pericycle tissue and above the protoxylem forming a complete and continuous wavy ring. Means secondary growth is also seen in the dicot roots due to the presence of a cambium. Okay. So, later on it becomes circular, and further all events are similar with the dicot stem. So, whatever the events which takes place, so same. Okay. There are two types of vascular cambial cells. What are those two types? Fusiform initials and ray initials. These fusiform initials gives rise to xylem and phloem, but ray initials gives rise to medullary rays in the secondary tissue. Medullary rays in the secondary tissue. So, here epidermis, endodermis, pericycle, these are the three consecutive layers, one after the other. Outermost epidermis, inner pericycle, then first primary phloem, then during secondary growth secondary phloem is produced, then primary xylem, and during secondary growth secondary xylem is produced. Okay. So, like this growth takes place. Okay. So, this is a secondary growth in dicot roots. In monocot roots and monocot stems, the secondary growth is absent. I hope you understood, children. Thank you for watching our video. Please subscribe our channel and press the bell icon for receiving latest updates.